Welcome to the next video in the Patterns in Nature topic. This video will be addressing the dot point, perform a first-hand investigation using a microscope to gather information from prepared slides to describe the sequence of changes in the nucleus of plant or animal cells undergoing mitosis. So we've recently been looking at the sequence of events that take place in mitosis, in particular the four phases of prophase, metaphase, anaphase, and telophase, which make up mitosis. And then we have the resting phase, which is interphase, and our final phase, which is cytokinesis. So if we have a look at slides that have been stained using an appropriate stain for that particular organism, we should be able to see how the chromosomes act in each of those stages and then be able to describe the sequence of events. So to do this, we're going to carry out the investigating mitosis under the microscope. So the aim of this investigation is to observe chromosomes and ultimately cell division through a microscope. So just a little bit of background information with our introduction. Chromosomes are only visible under the light microscope during mitotic division, so during that process of mitosis. They are most easily seen when stained by dyes. So dyes obviously color the chromosomes in order for us to be able to see them under the microscope. Okay, so during the process of interphase and cytokinesis, we can't really see the chromosomes, so we can assume that any of the cells that we're looking at that don't really have those visible chromosomes are in either one of those two phases, and the ones that are visible, we should be able to see where the chromosomes are located within the cell to determine whether they're in prophase, metaphase, anaphase, or telophase. So our hypothesis for this investigation, if actively dividing tissue is stained, then cells undergoing mitosis will be observed. So here we see an example of a broad bean root tip. Okay, so it's been prepared for us to be able to see the cells underneath a light microscope. So we can see that they've used a dark purpley blue stain, and we can see quite obviously that there are cells in different phases of mitosis. So here, for example, we have metaphase, where the chromosomes are lined up along the middle. We have anaphase, where they're moving to opposite ends of the cell. Okay, we're a little bit trickier to make out some others. This is probably late telophase, where we're starting to get that division into two new cells. Same here is probably between anaphase and telophase. But if we have a look at this slide here, this slide shows some really great examples of cells undergoing the different phases of mitosis. So as we can see, in those cells where there's no real indication of the chromosomes being visible as those stringy dark patches, okay, we can assume that the cell is in interphase. So majority of the cells at any one time are going to be in interphase, because if you recall from the video looking at the process of mitosis, majority of the cell cycle is within that interphase stage, getting prepared for mitosis to take place. So then we have prophase, where we can now see the chromosomes being visible, and they get ready then for the next stage, which is metaphase. We can see here that the chromosomes are lined up along the middle of the cell. Then we have anaphase up here in the top corner, where the chromosomes are pulling apart from one another. And lastly, we have telophase, where we have our division of our chromosomes, and we have the creation of our new, in this case, cell plate or cell wall plate, because they're a plant cell that we need obviously to create a new cell wall that runs through the middle of the two cells in order to create two brand new cells altogether. Okay, so a little bit different to animal cells, but we found that looking at onion root hair cells, or from the previous slide, broad bean root hair cells, we can really visibly spot the different process of mitosis. And if you recall, the reason why we use root tips is the root tips are continuously growing as the plant grows. Okay, so the root is continuously pushing into the ground in order to keep the plant alive. So therefore, it's obviously going to be undergoing mitosis quite a bit. So by having a look at the cells under the microscope and having a look at what's happening in each of the cells, you would now be able to describe the sequence of events that takes place during the process of mitosis and therefore meet the requirements of this dot point. And that brings us to the end of this video. So thank you for watching.